Welcome to Educon. In this session, we'll learn about mitochondria. Mitochondria are also called as powerhouses of cell. And mitochondria are not easily visible under microscope. Okay, it needs we can see mitochondria in electron microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. What is physiological activity? Like movement of ion, transport of molecules, and endocytosis, etc. In terms of shape and size also, considerable degree of variability is observed. Typically, mitochondria, they are in sausage shape or cylindrical, having a diameter of 0.2 to 1 millimicron in length and width, and average is 0.5 millimicron and length of 1 to 4.1 millimicron. This is the average size of the mitochondria, and each mitochondria is a double membrane bound organelle. So we have in mitochondria, the membrane surrounding the mitochondria is dual membrane. Okay, remember this. Where we are saying the double membrane bond, in nucleus also there is a double membrane, means dual membrane present in the nucleus. Where in mitochondria, the outer membrane and the inner membrane divide its lumen distinctly into two aqueous compartments. Okay, now you can see in the image here, we have a outer membrane and this is the inner membrane. So here this is the outer compartment, the space between the outer and inner membrane, and the space inside the inner membrane. So what's happening here? Outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments: the outer compartment and the inner compartment. The inner compartment is filled with dense homogeneous substance called matrix. Remember this. Okay, matrix is filled in the inner compartment. So this is you can see in the image, we have a outer membrane, the inner membrane, and inner membrane is filled with the matrix. The outer membrane forms a continuous limiting boundary around the organelle. This is the continuous, this outer membrane, it is a cross section of the mitochondrial image here. So the outer membrane is continuous, like it is not having any kind of partition, so it is continuous boundary, limiting boundary of the organelle. The inner membrane forms number of foldings called cristae. Now don't confuse cristae with the cisternae. Cisternae we saw in Golgi complex, remember that. Okay, here cristae, if it is more than one, singular is a crista, and the inner membrane forms a number of infoldings called cristae towards the matrix. Now the matrix it is here, so it is towards the matrix, that there is a folding. The inner membrane fold itself into the matrix, forming cristae. The cristae increase the surface area. Now remember from the previous classes where we saw plasma membrane also, there is some organelle in bacteria where plasma membrane, the infoldings of plasma membrane we found in bacteria, those are called mesosomes. So mesosomes are present in the form of sacs, vesicles. It increases the surface area of the plasma membrane. Remember that two functions are common here. The increase in surface area is observed here also, because they fold into matrix and increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. So the outer membrane, they have their own, it is having its own enzymes, and inner membrane also it is having its own enzymes in maintaining in coordinating mitochondrial functions. Mitochondria are the sites for aerobic respiration. Okay, they produce cellular energy in the form of ATP, hence they are called as powerhouses of the cell. And apart from all this, the matrix also possesses single circular DNA molecule, a few RNA molecules, ribosomes, and the components required for the protein synthesis. Okay, now during the start of this unit, in overview we studied about the membrane bound organelles and non-membrane bound organelles. Like ribosomes is present in both prokaryotes and eukaryotes. I told you that ribosomes are also present in some other organelles, membrane bound organelles, chloroplast, mitochondria, endoplasmic reticulum. So here is the example. The matrix possesses single circular DNA molecule, few RNA molecules, ribosome 70s. What is common? Now here you can extract questions such as the common between the prokaryotic cells and the mitochondria. Presence of 70s ribosomes, presence of 80s ribosomes. You can have multiple options. So in prokaryotic cell we see 70s ribosome, and also we see the same ribosome in mitochondria also, and it also contains the components required for the synthesis of proteins. And here the mitochondria divide by fission. So these are all the important points. Okay, you need to remember all this. Here powerhouses, it's an easy question, so mostly it is very easy, it's a straight question, so mostly you will not get, but you will get like the mitochondria possess single circular DNA molecule. Okay, and also it contains, you can have the questions such as, you can extract questions like mitochondria contains 1 circular DNA molecule, 2 RNA molecules, 3 ribosomes, 4 all of the above. So answer should be all of the above. So don't confuse, we need to remember all this. And mitochondria divide by fission, this also you need to remember. Okay, and with this we conclude this mitochondria, and we'll be having a separate video for plastids. Okay.